All right. Good morning, everybody.
Good morning.
Yes, that's the way I like it. In the elementary school, you want all the kids to say good morning. That's what we need. And it is a bright and nice, not too rainy a morning, but it's a nice morning. It's not too warm yet. And so it's beautiful. Welcome to the fall seminar series of iCubeCard. And she wants me to move back. They can't see me online. Okay, so online, guys, I repeat it. Good morning to all of you. So welcome to our fall seminar series. I'm Ranu Jung. I'm the founding executive director of iCubeCard, Institute for Integrative and Innovative Research. It is my pleasure to have Dr. Tatiana Wolfe as our starting speaker for the series. But before I introduce her, let me tell you, if you have not already heard a little bit about our institute, that the institute has got a mission to be able to make impactful societal impact. And how do we do that? Through a convergence approach that brings in different sectors together, the academic sector, the industry sector, the clinical sector, all together to find solutions to wicked problems that cannot be solved easily by one group alone, but require this kind of convergence to do that. And in the process, we are thinking about innovative solutions, research, innovation, entrepreneurism, so that those solutions, those outcomes that we can get to don't just stay in a lab, don't just become a pattern, but they can actually get deployed. And that uniquely defines us as an institute. So we look at 5 words. You have integrative, you have innovative, you have convergence, you have got societal impact, but then we want our solutions to be at scale. So that's what our institute is all about. And as we are doing this, we've picked up a grand challenge model, and the grand challenge is integrative health. And that is bringing, the way we look at it is bringing the food sector, health sector, and cyber sector all together in order to be able to promote well health law. And I just came back from a meeting in DC, and one of the things we were talking about is what is the future of health? And we talked a lot about it, that it's not just about cures. It is about both the prevention and also if something happens after disease. And so one of the things I had an opportunity to present was how we are looking at it. Making sure that we are moving everybody, keeping well health or moving people back to well health. And something like the imaging that happens, something like tools that are there, these advanced tools, they all help us in that particular aspect of that bigger ecosystem of moving us all towards well health. So Dr. Tatiana Wolfe, who's our guest speaker today, specializing in investigating brain mechanisms and the underlying clinical responses to diseases that are neurologically related or psychiatric, but also those might come because of aging. So perhaps as part of that would also be assessments of how we are doing a good aging and healthy aging that is going on all along, and studying to the people as they age in a manner that is actually important. Talk about health span, not just lifespans. So well health and how that might happen even earlier before you might see consequences of diseases. So she aims to generate new knowledge, and she's a medical physicist. So she's looking at not just applications, but she's looking at technologies, the parameters, the algorithms, the protocols, all of that actually allow so many others. I see Dr. Baran from from the psychology and sociology background. People who are application people who want to study need the medical physicists to be able to set things up that we can actually use that. So this is the bringing together of the tech world and the applied world. And she's looked at cancer treatments, amyloid removal therapy for Alzheimer's. She's looked at transcranial magnetic stimulation for treatment of resistant depression. So this is bringing in another technology built along with the MRI part, right? And then also studies that are related to multiple sclerosis. Some of you might remember that we had a speaker that came from CU Anschutz about a year and a half ago, and she talked about multiple sclerosis and all the issues with the loss of myelin and studies that they were doing both in animal models and in adults. So here is a very important diagnostic capability that MRI brings in the study of myelin imaging. And I understand that you have a lot of patents that are associated with in particular for myelin imaging and neurodegeneration. Dr. Wolfe was educated in the Department of Physics at the University of Sao Paulo. She completed fellowships at the MD Anderson Cancer Center and the Houston Methodist Hospital, and did a lot with myelin imaging. She has held research roles at Ohio State University, the Nationwide Children's Hospital, and now she's serves at the UMS Brain Imaging Research Center in Little Rock, and she's an assistant professor there. And she's the technology director of the Winthrop Rockefeller Cancer Institute 7 Tesla study. She's already has a collaboration with Professor Josiah Leong here. She's already given us extensive input and help and support in setting up our own CMA-X MRI system here. So we see this as a long, very fruitful partnership. So it is quite appropriate that we are starting this series today in the new building, now with new capabilities coming on board, that we have our very own state University of Arkansas Medical Sciences professor, Tatiana Wolfe, joining us. So I'm going to let her start. And before I finish that, I also want to tell you we have a wonderful speaker series for fall. Go to the website, and those who may be listening in it's i3r.uark.edu, and there's a whole list of speakers there. And then the next one on October 3rd is going to be Dr. Hyo-Young Jeong from UC Davis, and he will be talking on personalized digital healthcare, wearable bioelectronics for health monitoring, diagnostics, and therapeutics. So we're keeping the theme of image people, wear technologies, find out what is happening, well health, and really welcome back. Thank you.
Wow. Okay. Good morning. My name is Tatiana Wolfe. I am honored to be here. I'm amazed by the people and the infrastructure in the IQR. I've heard of the Institute, but seeing it firsthand is really inspiring. Thank you. Thank you for having me. And I'm really excited to join this community. It's a very special community, very interdisciplinary. I'm learning more and more about all the amazing things that are happening here. And I am excited to share the talk with you today on advanced magnetic resonance imaging and spectroscopy interventional clinics. So I am a medical physicist, and this is where my research lies. So I'll touch a little bit on the different things that we do. Here's a brief outline. So I'd like to share a little bit about my work in medical physics, and then talk about the significance of advanced MRI. And then we'll talk about what advanced MRI is versus conventional radiological MRI, and some practical considerations. And I bring this to share because I think you have interest in understanding some of the practical considerations for the IQR. I chose today to talk about 2 applications. I have actually 4 small funded projects to study 4 conditions, but I chose this 2 today: the Alzheimer's disease and depression application. And then I'd like to also discuss with you, bring forward the problem of the linear error model in application of advanced technology in clinical research and clinical practice. And then I have some fun features in the end. I am a graduate from the University of São Paulo, Department of Physics, University of São Paulo in Brazil. I'm very proud of it. I have a bachelor's and a PhD in math physics from the same department. I was in that department forever. And then I came to MD Anderson to do part of my PhD, and it was a great time to be there. And I came for the first time in 2010, then back in 2012, and I was there until 2015. So what I saw there at MD Anderson was Dr. James Allison got a Nobel Prize. We had Watson giving a lecture on the DNA structure, like things like that. It was a great time to be in MD Anderson, and there's a tremendous facility for imaging. It was paradise. It was great. Then I went on to do a postdoctoral training, a postdoctoral fellowship with Dr. Philip Corner at Histomatis Research Institute. I saw it built from the ground, and when I joined his lab, the question at the time was, they work in very effective interventions to grow functional nerves and functional new connections in the spinal cord and the central nervous system, but there's no way to monitor it without being invasive, without doing electrophysiology directly to the spinal cord, or there wasn't a way. So we worked together. He gave me the chance. He brought in a physicist to a neuromolecular lab, and we worked together to develop some things. We did. I love this piece of work. Signal isolation MRI is, but it's actually very simple. It's a technique. It's analytical solution to isolating the T2 starch signal that forms in this water pockets here. Do you see my mouse? No. I'll point. And this water pockets that form when myelin detaches from the axon. And that process is one of the first things that happen, one of the first alterations that happen when a neuron is dying or begins to die. It is the interesting part of measuring this is that it's a repairable process. So you can see it happening and you can see a recovery from it. And we worked on developing a signal isolation technique to do that. And we've worked on other things. I have worked with the group, the neurosurgery group, on an endotracheal neurostimulator, which is still, very successful, moving through these steps, with the FDA, working along back and forth. So it's been a long journey on that. Those are my disclosures. I'm not talking about any of this today, more than I did. This is where I am right now. So in 2023, I joined UAMS, the Brain Imaging Research Center, and Clint Kiltz hired me. For those of you who know him, he's fascinating, and he retired last year. We have a new director who's a great colleague, Andrew James, and the Berg functions really nicely. You guys have in fact been there. It's a great place to be. And I believe you guys are in the highest part of the state. That's the area there, and the difference is tremendous. I love it. The weather here is better. I am also a scholar of the NIMH Advanced Research Institute for Mental Health and Aging. It's been a great experience. I highly recommend people applying for that. The fellowship it's a 2-year scholarship, but like they say there, and I take it serious, it's eternally. There's no, once you are an ARI member, you are an ARI. And that's how I feel. It's amazing. I have collaboration and mentorship from top people across the country. It is a really good place to be. Now I'll move into my research focus. I have been recently, the past few years, really excited about brain metabolism and advancing quantitative imaging methods to help to help medicine. And this, there are over 90 million MRI exams performed yearly, with about 40 million just in the United States. And unfortunately, small fraction is actually to monitor what is happening with the patient upon intervention. Majority of it is diagnostic MRI, diagnostic imaging to define normalities from abnormalities, and very little is actually utilized. I believe one of the reasons that contributes to this limitation, this limited use of this amazing technology, is the lack of quantification. So it's truly a methods problem, not so much a engineering problem. There are great solutions, great hardware, but we need to have methods for precise verification of things that come out, signals that come out of the MRI. And as we learn from this famous painting, I've seen this in many, many medicine lectures from Samuel Fields, the doctor is super vulnerable. He doesn't know whether the patient will get better on intervention. The father is vulnerable. Maybe perhaps you didn't even notice the father in the background. The mother's crying. Everyone is vulnerable. The patient's vulnerable upon intervention. It would be very nice to interact and help this problem, which persists nowadays. And Jane Moore is, she's controversial, but she's teaches in this article. She actually published a scientific manuscript about this painting recently to discuss that medicine is not just a technical practice, but a human practice, deeply human interaction. And I believe physicists can contribute to this environment by using technology to connect the patient to the doctor and the doctors and the family. And that's how that's the value of the profession, I believe. Now, what exactly is advanced imaging and how is it different from conventional radiologic MRI? I'll tell you that there are two major imaging conferences in the world, the RSNA, Radiological Society of North America, and the ISMRM, the International Society for Magnetic Resonance in Medicine. And that is the difference. So the RSNA is the radiologists conference. Kerry is shaking his head, and the ISMRM is the actual MRI. So advanced modalities are modalities that will process the signal from the MRI before generating a meaningful image. So the images that come during, say, a functional MRI position, they don't have any meaning in isolation. They only make sense when you collect a temporal series of them, process those temporal series. What does process mean? It means apply statistical models, mathematical models, and then statistical models. Then you generate a meaningful map of something, some quantification of something. In this case, in the case of functional MRI using bold technique, bold contrast, to say you generate data about the connectivity of things. How are things functionally connected? So you go from time series of blood deoxygenation to a map that overlays into the anatomy and tells you about brain function or likelihood of brain function. Another advanced modality that also requires processing of signals is diffusion tensor imaging using diffusion-weighted techniques. So diffusion-weighted image is widely used in the clinic. It is a snapshot of how much, what is the diffusivity of the water content in the brain. And I should disclose, I'm only talking about the brain today, but these things have applications all throughout the body. I'm going to focus on the brain because I love the brain. But diffusion tensor imaging in the central nervous system measures the diffusivity of the water, so it acquires data, a temporal series of water molecules moving in space in response to the electromagnetic field applied to that area, to those cells. It is specific to the water, to the intra-axonal water, not everything else. Everything else adds noise, but the quantity you're trying to model is only that intracellular, intra-axonal water diffusion. And in order to do that, you apply several mathematical models, and then you can trace connections in very specific brain regions. This paper is one of my favorites. It's Josiah's paper, and it is a fascinating piece of work. He talks better than I can ever talk about it, obviously. But that's how you discover connections in the brain is using advanced diffusion tensor image, so you can put tensor, mathematical tensor, which is based on likelihood of diffusivity in the cell. So it's all ultimately mathematical models and statistical models to get that. This is my new favorite advanced technique. It is PRESS, so point result spectroscopy, using an additive pulse. So MR spectroscopy, NMR in the body has been done for decades. I think it was done before image was used, because spectrometers are easy to understand. If you have a degree in chemistry or physics, it's easy to understand, and you can infer about the molecules in the sample. But only recently, very recently, editing pulses were able to successfully remove signal from predominant molecules. So NAA is and acetyl aspartate is molecules everywhere in the brain, more or less evenly distributed. So it doesn't tell you much because it's everywhere equally distributed, unless you have abnormalities. But if you're trying to understand how the regions are connected or function, it's not a very good mark. So you want to get rid of that, but that's huge peak in your signal. So editing the pulse at the acquisition level, so flipping the NAA J signals or spins allow you to see the other peaks. And those are peaks of interest in our chemistry. GABA and GLX stand for glutamate and glutamine together. It's harder to separate them in 3 Tesla. You can separate them in 7 Tesla generally. But in 3 Tesla, this is how they look like. They look like 2 little, then call it GLX. And this is about brain or cellular metabolism, and this is a neurotransmitter. So you can measure very, very nice things, but it requires post-processing of that spectrum. So it's an advanced technique. And then you get really meaningful papers, and also you get to apply to problems of the mind, some problems that don't have psychiatric conditions, that don't have frank remarks in the anatomical MRIs. Because now you're talking about brain neurochemistry. And no contrast injection, it's not ionizing radiation. It's pretty fascinating. Let me tell you about why this is translating into clinical practice right now. It's still pretty novel in the research realm, but there is so much advancement in automation. Acquisition is actually really short. The edited spectra scan, for example, you can do that in 5 minutes, whole brain neurochemistry map. It is pretty fascinating. And you have the machine to do things like that, by the way. And I think that is critical to determine the feasibility of doing this in patients, and in patients who have conditions, who move, who are extremely uncomfortable, who are in pain. It's a whole different set of a different population that requires adjustments to the scanner, and that allow for application in different things. I do have a project on where we are looking into brain metabolism and perfusion levels in women or breast cancer patients who have received chemotherapy several months ago, and now we're looking into their brain neurochemistry to see to see the trajectories, how myelin synthesis is affected, how connectivity might change. But I'm not talking about that today. I'm going to talk about the Alzheimer's disease and the psychiatric patients. The reason why I'm able to study all these things is because the BERC, the Brain Imaging Research Center at UMS, has launched a new program where we're offering now these modalities. Currently, these are the modalities being offered as clinical exams. So now these patients that require hyper-personalized assessments are coming to our research center. And then we wrote IRB protocols and we have authorization to do research on them if they consent. So we can invite them to research at the time of their MRI appointment. And that gives us access to the population. So I do have a postdoc, Ashley Pike, who works with the MS cohort. And the amount of data is tremendously high. It's easier to collect data this way because they have 100 patients a year coming for these exams. But I won't talk about the lesions today, but I'm happy to talk more about it if anyone is interested in that topic. There are some limitations. One huge limitation is that analysis softwares that you can use for clinical cases is very limited. They have to be FDA approved because we are in the United States, and there's only a few of them that exist, and they're very expensive. We are currently using the one that Siemens has because when you buy the scanner, it comes with the scanner, and this is something important to have. We were talking about this earlier, and if you do have, you can offer these things clinically, and also there's a lot of motion and real world problems that you need to solve. Like these pillows are really important and it makes all the difference. When you're talking about precision quantitative MRI, the sample cannot move. If it moves, everything you're saying doesn't lose its meaning. So you really need to make sure it's almost, if we could, we should do stereotactic imaging, which you can't, because you can't really, put a hardware and keep people's heads still. But you put nice pillows, and that helps a lot. And I should say that this program, this hyper-personalized MRI services, it's an initiative who Dr. James pushed forward. He pushed forward this at UAMS. There is a lot of behind the curtains work that goes into establishing CPT billing codes for each of these advanced modalities. Working with the neuroradiology team to understand and read these images because they are different than their conventional MRIs. So there's an educational piece to that. And then creating reports that can be read by neurologists, by the prescribers, by the neurologists, the neurosurgeons. So it's a whole integrated loop, and it touches on both education and technology. It's not just the technology. My job is just technology, but there's a whole lot that goes into this program. I love this image. This is we have students in there. Can you tell what this is? This is it's an MP2 range image, and this is the first inversion after the first pulse and the first spin. We have an IR, inversion recovery, and this is the first image generated. This can generate 3 images. This is the first one, and you can see the central medial nucleus of the thalamus in the contrast, and it sounds such a specific thing, but it is the place in the brain where neurosurgeons implant deep brain stimulators. It is what's called the mirror in the brain. So it is a place where epileptic seizures go through 100% of them. So if you put an electrode in there or a series of them, you can create a refractory circuit to prevent the seizure from spreading further. So but it's in this small neurofield in the thalamus, and it's not in the neighboring neurofield, it's only in that. So you need to be able to see it. And we're doing this at UAMS, and early 2024, we worked with the neurosurgery team, Dr. Victor Pallis. He performed the first DBS implantation in the state of Arkansas. And now there has been over 20 of them. But because of this image was not created by me. It was coded in the machine by I did, but the image originates from a 7 Tesla version of it that exists at Harvard and MGH. So this is just an adaptive versioning of it, but it can be run in the 3 Tesla scanner And I brought it here for you, so you have it too. Now, this is the overview of the projects. I don't think I have time to talk about the breast oncology project. I will, I'm really excited about both of these. So the first one is, and these are applications in interventional clinics. So these are patients that come to our scanner during their treatment. Let me tell you about Alzheimer's disease quickly. It is a devastating disease. It affects over 7 million Americans, but look at this, over 12 million caregivers. So it is an enormous impact on society. And there has been a newly approved, FDA-approved drug to delay progression of cognitive impairments in people at risk for Alzheimer's disease. That drug was approved recently. I have the dates in the next slide. And that drug was approved because it's safe. So there's an open label clinical trial showing safety. And it was approved because it removes amyloid at a chemical stage where before it forms a plaque that is then sticky and can't be removed. So it's a very interesting therapy. Why it delays cognitive impairment is still unknown. As far as I remember, amyloid has nothing to do with neurotransmission. So how removing amyloid prevents changes in cognitive decline, right? Okay, here we go. It was approved in January 2023, and reimbursement by Medicare was approved in July of the same year. And this is the treatment cost. And it is remarkable. The difference, the effect of lecanemab therapy in cognition is remarkable. So it literally delays the progression. So patients who come eligible for this treatment, they have confirmed amyloid pathology. That means they receive a lumbar puncture to test their CSF for the presence of amyloid. So they have confirmed amyloid pathology. And then if they would like, and they have genetic markers tested, if they would like, they join the therapy. And that takes UAMS between 12 and 18 months. And during that time period, they receive 7 to 9 MRI exams to monitor something that I will talk about in a moment. But the principle is the removal of amyloid. Sometimes during the removal of amyloid, there is little edema that forms. Sometimes an active hemorrhage, microhemorrhage is called, or sometimes it's not active, but it forms an edema. And that is called ARIA, amyloid-related edema. It's radiologically visible in some exams, so you can pinpoint where it is, but we don't know why it happens. This is how we monitor ARIA. We run an advanced modality called SWI, susceptibility weighted imaging, and which is a calculator. So it's advanced. You collect gradient echo data and you compute these maps. And and if there are microhemorrhages, you see this black dots that show up on this side are microhemorrhages. And there's criteria, there's radiologic criteria. So the Alzheimer's Association came out with a consensus of how to quantify ARIA. And that's the radiologic criteria, the radiologist's job. My research is now focused on understanding how exactly removing amyloid changes brain function in this population. The primary hypothesis we were proposing, so this grant is under review, everybody fingers crossed please, October 19th. And the primary hypothesis is that by removing amyloid, there is a change, a direct change in metabolism. So the neurons can now communicate better. So I fundamentally think, I didn't put this on the grant because I don't think it would sound nice, but fundamentally I think that there is a dielectric change. So when you have amyloid plaques, you have a dielectric between cells trying to talk to each other electrically. When you remove that, you have a better electrical environment. I think that it's that simple. But because they communicate better, we should be able to see alterations in glutamate and so GLX alterations. And I collected some preliminary data, and this is very interesting. So the IRB protocol, like I said, these patients come to our scanner 7 to 9 times throughout the therapy. And they come initially, so I have a baseline scan on every patient, which gives us this, they come for clinical reasons, they come for rule out presence of stroke, tumors, and other things that they could have comorbidities that would prevent them from receiving therapy, preclude them from receiving therapy. But if they enroll in our research protocol, I also collect molecular imaging. So I collect that edited scan on them. And this is a baseline, this is an example. And as you can see, tissue's clear, anatomically clear, radiologically clear. This patient went through therapy. 3 months later, came back with something like this. This is ARIA, this is the amyloid-related immunotherapy. And we have observed a molecular signature, which wasn't at baseline. It's elevated myonostal. Myonostal is a molecule related. I'm showing you myonostal, but we detected other things altered. It's one patient. So I need a bigger study to understand how we can actually help this problem. But I'm saying this because this is beyond my primary hypothesis of energy alteration. This is about the mechanism of ARIA formation. So multimodal MRI can give us a lot of understanding in practical ways and in mechanistic ways. Here is the example. We do a very fine job, Leslie. We do a very fine job trying to apply correction factors, so tissue density correction factors, to this molecular quantifications to try to separate what amount to truly quantify what amount of metabolite comes from neurons, so gray matter, versus non-neural cells that are tissue with predominant glial cells, so white matter, and myelin. So this ones I want to call myelin-related metabolites, but we can't. So I pretend that today I can. So I want to truly quantify the myelin-related metabolites, and then so we do use diffusion tensor image to compute the fraction of white matter in the MRSI voxel to then apply a correction factor to the molecular measurements. So it's quite multimodal. And we saw alteration of glutamate as well. But we saw glutamate actually went up from 0.5 to 1.243. So huge increase in glutamate 3 months into therapy. I think this supports our theory. And we collect a battery of neurocognitive data in these patients. That's not my expertise. I have a great colleague who deals with that, but we do collect several questionnaires and a lot of neuropsych data, as well as a couple of tasks. We collect a cognitive flexibility task, the response card sorting task, and the flanker, so the inhibitory task, and I think that's it as far as tasks. We also collect rest state data. We're doing all of these projects in the open science framework, so the idea is to generate database and study protocols that can be replicated in other centers and add on to data sets and to knowledge. That's one application. The other application I am in is this one. So in people suffering from major depressive disorder who have not responded to medication or to other interventions, there's now a new hope. And I say this because it's truly impactful. There is a modality, a protocol of TMS. TMS stands for transcranial magnetic stimulation. It is essentially a chair with a coil that will be positioned, a coil, I mean, electromagnetic coil. So there's an actual electromagnetic coil there, positioned in somewhere in the brain. So not I should say in the head. So it's non-invasive. And that will cause small magnitude but high frequency stimulations to the neurons in the region near the electromagnetic field. And that stimulation propagates throughout the brain because every region of the brain is virtually interconnected to every other region through some pathway. Virtually, I said, don't look at me like that. I was like, no, you're wrong. No, it is virtually through some pathway. There's a connection. So it propagates through the brain. And that effect has a functional response associated to it. So you stimulate here and you may get a functional response elsewhere because it's part of the circuit. And that's the principle of TMS. What is novel about SAINT TMS, SAINT stands for Stanford Accelerated Intelligent Neuromodulation. They made it fit into SAINT. That modality is innovative because it is an intensive protocol, very intensive protocol. So a patient comes in, there's a baseline MRI, I'll talk about it in a minute, but the treatment timeline is 5 days only. I should mention, TMS is typically several months of treatment and with the flip of a coin, in response. So it's a hard decision for the patient to engage, especially since they are severely depressed and they haven't responded to medication. Now, SAINT is 5 days of treatment, starts on Monday, ends on Friday, 10 hours a day for 5 days. And patients in the TMS chair on and off, 15 minutes on and off throughout the day, the treatment day. There is a baseline MRI I mentioned. The baseline MRI is to identify in a hyper-personalized manner where to stimulate. So my point might be different than yours and so on and so forth. And we find that point by computing the functional connectivity of the location, the voxel of highest, strongest functional connectivity between the dorsolateral prefrontal cortex and the subgenuine cingulate cortex, which is the functional target you're trying, deep brain target that you're trying to stimulate. So if you could go in there and stimulate that area, you would, but you can't because it's in the middle of the brain. So you rely on functional connectivity networks and for the stimulation. It is absolutely remarkable. The open label trial for SAINT showed over 86% remission rates, which is unforeseen for any therapy, any drug that has ever existed in mental health. And this is fascinating. So we're trying to replicate some of these numbers. There's a difference between the study I'm going to show you and this open label trial. The open label trial was done with adults, and my data is only in older adults. That's one major difference. And there's other differences. I'm happy to talk more about it. But we're trying to replicate some of this, and we're trying to understand what are the molecular changes that are happening while the stimulation is taking place. Here's the functional target, the SJCC deep in the brain. It's the central slice of the brain, and it is a classic circuit in depression. It's not new. SAINT, I should say, after being FDA approved. Did I have the numbers here? Did you see how much it costs? Yes, so this is the cost of the treatment, and Medicare reimbursement has been approved. The functional connectivity between the left dorsolateral cortex and the SGCC has been tested as a single predictor of response to SAINT TMS. So it's really a strong connection, and it works. This is how it works. It works through the function, through modifying the functional connectivity of the. I haven't used this. Modifying the functional connectivity of between these two regions. And they're anti-correlated. So if you stimulate one, you suppress the other one, roughly. But why does it work? You change the function of connectivity. What does that mean? You're changing blood consumption by these neurons. What does that mean? Why does it work? We then launched the Mars project, and the Mars got funded by the Medical Research Endowment to UAMS, and it's a pilot, and hopefully it will be our one will be funded to expand. Two main hypotheses are that first, the structure matters. We believe that you need to have integrity throughout the circuits of the brain for you to be able to propagate this functional effect throughout the brain. It sounds very simple, but it has not been tested. So we think that the structure matters, the neurostructure integrity matters, and you can guess by now that I'm going to use diffusion, tensor imaging to look at that. And the hypothesis two is about the depression circuit. So the level of GABA. GABA is an inhibitory neurotransmitter, and it is, and we think SAINT is modifying the levels of GABA in the functional target. And because of that, you have a antidepressant effect. It's important to understand the mechanism. This is a mechanistic question. And it's important to understand this mechanism because if that's the case, you want to control the medications, the GABA-related medications that patients are taking before going to SAINT. And you also want to potentially measure GABA levels throughout therapy to titrate therapy, dose, frequency, duration. Right now, the protocol that the FDA has approved is set for everyone. And some are responders and some are not. I'll show you some data. This is the demographics of our characteristics of the subjects that have enrolled in our study. Not all subjects, less than 50% of patients receiving SAINT have enrolled in our study. We still have we have difficulties recruiting. They are depressed, they don't want more in their lives when they come in. It's difficult to recruit them. But they're older adults and majority females, I should say. And this is very important. Most of them have normal cognitive function. And some of them have mild cognitive impairment. These are the individual trajectories. These are the individual trajectories of each of the patients. And I only have a few here, but that's the figure I had. What is interesting is that some patients rebound. Okay, the y-axis is the GDS-15 scale, is the geriatric depression scale. So this is just the clinical outcome. And some patients respond throughout, but some patients rebound and then respond again. And some patients simply rebound and go high and return to the depressed state. I can talk more about each piece of this. I ran a correlational tractography. So we traced the tractography of the entire brain. And then we took tract by tract, put in a statistical model to see which ones, the integrity. So I'm actually quantitative correlating is keyway, quantitative anisotropy. It means it's a function proportional to the integrity of the fibers. So we run a correlational model between the whole brain, so any tracts, how do they correlate to the outcome? And we found one that correlates to, that's predictor of outcome. It is the DRTT. It's a pathway, it's not a single tract, so it involves several tracts, and it's a brain pathway. It has been reported, this is how the DRTT looks in the Human Connectome project data, which is an average across over 1,000 subjects. And this is how the DRTT integrity looks in our sample. So it is different in morphology and in QA. It's scaled, color scaled. So see that. But here, this is very interesting. This is a responder, the DRTT responder versus a DRTT pathway in a non-responder, and they're dramatically different. So we have statistically found the tracks that could predict, and we see a difference in a responder versus a non-responder. I like this piece of work. But again, this is a predictor of therapy response related to circuit integrity. It does not explain why the treatment is effective. I think that question has to be answered by molecular spectroscopy. We did measure, so we used a sequence developed at Hopkins, which is also part of the HPCD protocol, the Isthmus, which is a dual echo press localized proton spectroscopy sequence, edited proton spectroscopy sequence that allows to measure GABA levels, among other things. And we have found that that potentially, in our limited sample, potentially SAINT may act to restore connectivity, shifting GABA metabolism down and aligning with clinical improvement. So that's the connection, the link between molecular, structural, and response upon intervention. Again, I'm happy to talk more about it. I just got a card that says 5 minutes. I gotta finish. I do want to talk about this a little bit. So the linear error model, there is all of these measurements that I'm doing. In order for those measurements to be used in the clinic, they have to come with an error bar. We have to tell the physician it's 3.2 millimolars of GABA plus minus X. We need to be able to say that. And currently, there are no mechanisms for that. So that is lacking. We need to work on this problem. There's several reasons why we need uncertainties, but I know everyone in this room knows the importance of uncertainties, many uncertainties and errors. This is a new toy I'm playing with. It's a really cool phantom. It has several vials with different concentrations of GABA and glutamate and choline, and we can use it to compare sequences and acquisitions in a more systematic way and try to understand what is the best acquisition scheme for a particular metabolite that you want to use or you want to investigate. So hopefully we'll build sensitivity curves in this phantom and publish those and use those in our models to estimate uncertainty. But it's a very important piece of work I'm currently working on. And that's it. I'm very thankful for everyone in the lab, for everyone who I still collaborate with, the Methodist and in the other places in Madrid. And I am very thankful for the lecture here today this fall.
Fascinating and new lingo for all of us and learning, especially with having the MRI here. We expect that there'll be more and more talks. And actually, next week, same time, we have Dr. Prabjot Kaur. She's a research fellow at Boston Children's, and she's coming here, and she will be giving a talk at 9 a.m. next week. Open to questions.
I've got one. So the study related to Alzheimer's that had, I think, either 24% reduction, you said it was a year and a half study. Is that the extent of the study, or is there ongoing? Is it only given drug during that timeline, or is it giving past that, and is that an ongoing study for long term?
No, that's the clinical trial data. So it was measurements during the medication period. So it's a 52 doses. 52 weeks treatment period and measurements up to 6 months more. And that's how they measured in their clinical trial. And that's what got them through the FDA for approval of the drug. In our sample, I have collected pilot data, and I only have data at 0 and 3 months because I don't have money to study subjects at 0 and 3 months. But what I'm proposing is to follow a whole sample throughout their treatment period as well. So this is during the drug flush, the amyloid flush out. Our IRB protocol does include the possibility of collecting data later and later time points to see about the long-term effects of lecanemab. I think other centers are looking at the long-term effects in terms of cognitive impact. I'm not an expert in neurocognition, and I don't. So my focus is not to look at that piece. Anyone is welcome to join us and look at that piece in depth. And I'm really interested in understanding how the heck this neurons go back to functioning if amyloid is not actually implicated in neurotransmission. But does that make sense? Yeah.
And fascinating topic. Sorry, you want to go ahead?
No.
And as we discussed yesterday, you brought me back during my PhD days when I used to do a lot of brain stuff. From my perspective, when we talk about amyloid formation in the brain, so that's due to a protein aggregation, right? And it can be any protein. So from my interest of biomarker discovery, like you have a fascinating tool where we can phenotype, right? Or see the early onset progression. So how about like what creates this protein aggregation more in this subpopulation? So from basic biology, what I understand is it's the chaperones and misfolding that leads to protein misfolding. So is there a possibility that when you phenotype these subjects and you know amyloid formation and better recovery, is there a possibility that we can detect the level of chaperones or mutation in this chaperones, and we should have the capability to do those things here as well in very near future?
Using in the brain, you mean in vivo in the brain, using.
Well, it can be just from the blood, we can just extract the DNA and see if there is a particular mutation we are looking around.
You know that better than I do.
So I was trying, since you're advocating for the molecular mechanisms as well, which there can be. So I was just kind of.
The onset is a big question. Onset of Alzheimer's is a question. I don't know. My hopes are that if we can understand if my hypothesis proves it right, if it is related to glutamate metabolism, then we can look at this pathway non-invasively at earlier ages and look would be ultra early stage Alzheimer's, in people with genetic markers, right? So we could apply that to prevention of progression. And maybe eligibility in the future, eligibility to initiate the treatment before confirmation of amyloid pathology in CSF, right? That's my hope in terms of contributing, integrating imaging to contribute to the field. Now, why these people you're talking about why they're at risk to begin with, right? What is their genetic miss?
And my interest would be like, you're flushing out the amyloid and there is a recovery. So then how long does it take for amyloid formation again?
Yeah, I don't know.
So,
That's not, I know that that's not known.
right.
Because the clinical trial ended and the data collection ended and that's it. And maybe there's people in New York trying to bring the same patients back to look at this later. Maybe there are, not aware of the studies. Unfortunately, not everything is pre-registered in science yet. So we don't know, but it's, very, very important. I think I missed saying this, there are 2 lecanemab centers in Arkansas, one in Little Rock and one here. So I'm really interested in tapping into this population.
Here is at UMS Northwest Arkansas or so?
No, it is a private clinic. I
Private
know
clinic,
one
okay.
there, but
Alright.
but I will get to know them.
Yeah, okay. Because that's a study that we could run
That's
an MRI.
that's the idea.
Ask about the spectroscopy and the temporal dynamics of things like
Yeah.
glutamate and GABA, and it seems you're taking a snapshot.
Yeah.
And you're taking a snapshot of things that I think are changing on fairly rapid timescales, and I guess part of it is, when you getting these measurements, are you measuring presynaptic availability of these neurotransmitters, or are you measuring?
It's a
What
great
are you
question.
measuring?
Everybody's asking that question since the inception of MRS. Here's the thing, I can tell you the theory. The truth, the theory is that, if you think of what we're actually measuring, the lines and the spectrum, they are, it's chemical shift in but the data is a part of frequency, right? So those frequencies are geometrical patterns in the molecule. So if a molecule is bound with something else, some receptor or anything, you would think that additional lines would appear in the spectrum, okay? And for that reason, the model, it's very specific actually. In this case, I'm showing you GABA, but you see three peaks. The green line is my model, GABA. My criteria of fitting, the curve fitting criteria, uses a three peak up approach. If I wanted to start differentiating them between GABA A, GABA B, bound, free, available, not available, you have to include in the model additional lines, and those lines will be at the north, and it's harder, and that's why they're not modeled currently. So the answer is, I don't know which GABA we're measuring. I don't know which one we're measuring. We're measuring the one that has best curve fit to the model, like the linear combination model function. And this is theory, and there's also the neuromolecular theory, which is if, say, GABA or say glutamate. I think you asked both. Yes. Either one. Yeah, they're both complicated equally. If you're looking at extras, if you think that you need the hydrogens in neighborhood to contribute to your signal, then we're more likely to be detecting extracellular molecules. So if they're internalized already, and so that's just inference of what stage of the molecules we're measuring, what population of the molecules contribute to the signal. But like I said, the truth, you have to run several mathematical models with several different lines and simulate the interactions in the different states, and then also simulate that in the condition of normality or pathology, and the presence or absence of things like amyloid, right? And we haven't. It's undone. It's not done.
So, but if you were to run the same scan 10 minutes later or half an hour later after you stressed them or had them do some activity, would you expect to get the same measurement if there's that kind of?
So I should tell you, it is normalized chromatin levels, which is, so chromatin is pretty stable in proportion to the number of cells, total number of cells in the voxel. So that's an attempt to correct for fluctuations that would be unrelated to the dysfunction, right? Because majority of the cells will not fluctuate their whatever levels, signal levels. So you have you can try to attribute the variance in the signal to the neuron in that voxel. So is that what you're saying? You have the confounding of, other cells are also energetic depending on what you're doing, right? Eating or not eating or whatever. And they all contribute to that glutamate signal, the GLX signal. It's true. But then we normalize by chromatin to try to circumvent that.
Yeah, Jimmy's question just piqued my interest about this. Have people tried task-based MRS? Like the early days of fMRI block designs, on-off some behavior?
I just came from this conference in Bangor, Wales, University of Bangor hosted. It is a functional MRS symposium. And yes, that's pretty amazing what's going on there. People are doing it. Acquisition takes time. So fMRS has to work with single acquisition. Currently we require 300 spectrums and we average them. I'm rounding to 300, but it's complex, right? And you do on and off and resonance. So it's a lot of data collected, so it takes minutes. these people are trying to do fMRS with single acquisition, lots of noise, with vast majority of the signal is noise. But then you have these models that will search for this very specific molecular signatures of certain things. And but yes, people are detecting variations of glutamate with task engagement and in a block design, much like fMRI. It's single voxel still, but it is really pretty. It's promising. I think it's the future.
Other questions?
And then you can create a theoretically perfect correction vector for what you're saying. Because then you have a condition with and without engagement.
No other questions, then thank you very much.